Hello, welcome to the meeting. We haven't had a recording in about 2 weeks, so I'm giving an update. Our original meeting today was pretty disjoint. People were joining at different times, so I didn't want to record from the original meeting. So I'll be giving an update on some things that are going on in the group. And then at the end, Karan is going to talk a little bit about some of the work he's been doing recently. Some really interesting things with genomics, next generation sequencing, and deep learning techniques. So those are two areas that are pretty intense. And so joining those together is, kind of a hard problem. He's also going to talk a little bit about the axolotl data set that we've talked about in the meetings before, and maybe finding some appropriate genomic data to integrate with that. So I wanted to talk about a couple things that are going on in the group. The first is that I prepared a blog post for Darwin Day, and I'll talk a little bit about that. We'll go through that. The second is to go through the GSOC, Google Summer of Code projects. We've had a couple of inquiries already for the Graph Neural Networks project. I wanted to go over that a little bit. So I wanted to talk about a special journal issue we're working on, we're trying to propose. I also want to talk about this. We have a new partner that we're going to be involved with, perhaps for Google Summer of Code. It's Toki. It's an open source project, and Krishna Katyal, who's been one of our contributors, he's actually working with them now, and he's going, they proposed a project, and they're applying for our master organization that we're applying to be a part of Google Summer of Code. So that's something that we'll go over. We'll go over Towhee and what it is, and then we'll go over this larger concept of anything to vec. And then we'll get into some papers, and then we'll talk, you'll see more on Karan's work. So let's get started. I'm going to share my screen here. So the first thing I want to talk about today is this blog post for Darwin Day. It's on Gerification, and we talked about Gerification, I think, in the past 2 weeks. So this is from a blog I run called Synthetic Daisies, and I've run this blog since the end of 2008. And there's a lot of content on here, like almost 400 posts over that time. And there's a lot of content here that's I've worked on. They're just blog posts, so they're actually not to be dismissive, but they can be pretty long. They get up to like 4,000 words. So there's a lot of good content on this blog. And so if people want to go through this blog, and there's a lot of content on development, on evolution, on computing, on general theoretical biology, a lot of different topics. So, if you go through this blog and you find a post that you find of interest, maybe you want to expand it and work with me on that, please contact me and let me know, because we can expand this out into a paper. There's also a GitHub repository or a GitHub site for Synthetic Daisies, which is like a recap of the greatest hits. So I can talk about that later after I talk about this post. So anyways, this is a post for Darwin Day, and I've been posting for Darwin Day since like 2014. Darwin Day is February 12th, I believe, and this was a little bit late, but that's okay. So this title is called Gyrification of the Tree of Mammals. This is a picture of old Darwin here. Usually we see young Darwin and old Darwin. Old Darwin is the beard, and young Darwin is younger, and I guess before he went on the Beagle versus after he went on the Beagle. So that's the idea. But basically, it's an opportunity for people to communicate different topics in evolution. So this year, I'm going to focus on the evolutionary origins and developmental emergence of gyrification in the mammalian brain. So gyrification is when the neocortex, which is this outer layer of the brain, becomes wrinkled. And so it often becomes wrinkled due to physical processes, but more often due to the growth of this outer layer in terms of its size. And it grows and grows, and it can't really fit into the skull anymore as a smooth surface. So you get these folds and wrinkles, what they call gyri and sulci, which are these areas that are like sort of still on the surface and areas that are folded inward. And so those, it basically changes the topology of that sheet, and it has effects on function. It has effects on it's on different aspects of it's actually, if you look at the different layers of cortex, you see that, sometimes if you have a developmental disorder like microcephaly, you have a disruption in how those sheets are developed, and so you end up with a smoother brain. So all these things are tied together, the development of the cortex and the folding and all that. So I went through a bunch of different data on this topic, differentiated between gyrification and lissencephaly, which is where brains have a smooth surface. So if you look at, let me see if I can expand this out a little bit so we can see. This is the brain of a rabbit, and this is the brain of a rodent. You can't really see the rodent, but the rabbit brain is a little bit bigger. And what you see is that it's very smooth, this cortex. So in fact, it's not even really that much developed. It's just very smooth, and that part of the brain is smooth. It's the frontal part of the brain. So in vertebrates, you have a three-lobe brain, essentially. You have the brain stem, and you have a middle section, and then you have the frontal section. Middle section is what in human brains we call the medial temporal cortex, which is where we have things like the amygdala and the hippocampus, things like that. The front part of the brain is this neocortex, which has six layers, and there's a very specific processing that goes on through what they call columns across these layers. And so we have that. In rabbits and rodents, and this is what we call an outgroup in phylogenetics, we have these smooth brains, but in the in-group, which is all of primates, we have various degrees of folding. And so you can see in some of the prosimians, you see very small brains, very small forebrains with a limited amount of folding. And then when you get into some of the monkeys and apes, then you start to see larger brains where the cortex is folded. So in older monkeys, in gibbons, and then in great apes and humans, it's a lot bigger and a lot more folded. And so this is all, this has genetic factors involved. This has morphogenetic factors involved. So, cell proliferation and things like that. And then you have these physical factors, which are, if you expand a sheet in a space, and I think we talked about this with crumpled paper a couple weeks ago. If you expand a sheet in size, if you crumple it up, you can restrict that size down, but you still have the same amount of space there. You still have the same amount of surface area. So that's why you get a lot of this folding. And so in this tree, you see that there are, if we trace these traits, what they do in a phylogeny is usually these trees are built on discrete traits. And so you can actually look, every branch represents a certain number of traits that distinguish that branch from another branch. So we have, in primates, we have these traits here: neocortical enlargement, visual, motor, and somatosensory system specializations, dorsolateral or prefrontal cortex. Those things appear in primates, and in the outgroups to primates, or the outgroups to some of the major primates that they have in this tree, they don't exist. And an outgroup is just a comparison from somewhere else in the tree of life. So people use outgroups to root their tree to figure out which traits are ancestral, which traits are derived. And so the outgroup is assumed to have some of these ancestral traits. They also have derived traits, but they shouldn't be anything similar to what you would see in the in-group, or the primate group in this case. So you have your different primate groups. You have another set of traits with anthropoids, brain enlargement, reduction of olfactory receptors, and trichromatic vision. These all appear at this branch. And then as you go on, you get to hominoidea, brain enlargement, reduction of olfactory receptors. And then they place the folding of the neocortex in this context. Okay, so then the second topic is to talk about, how Gerification may be a product of convergent evolution, not just simply as a matter of this sort of specialized evolution where there's what they call monophyly, which is where it's restricted to a single lineage or a single part of the tree. You see cortical enlargement throughout, actually, mammals. You see it in pilot whales, you see it in elephants, you see it in humans, and you can see humans are in the primate tree, in primate part of the tree of mammals. Elephants are in another part, and whales are in another part. So those expansions of cortex occur throughout the tree of mammals, and you have smaller brains also in between. So it's what we call polyphyletic, or it's evolved multiple times, maybe from the same precursors, maybe not. So we can look at different genes, we can look at other conditions that exist, but the idea is that basically it's a product of this convergent evolution, or what they call polyphyly. Okay, so but there are also, so there are a lot of physical constraints that can be involved in this. Another thing that we look at in evolution is we look at Elemetric scaling. So in this case, we have a bunch of species that are all lined up, and they have their brains. We have rats, lemurs, wolves, and humans. And we can look at those brains to look at the relative size of the brain, relative size of the cortical sheet, and this metric GI, which is the Gerification index, which marks, how much folding there is in the sheet. And so if we look here, we see that rats, lemurs, wolves, and humans are on this sort of continuum. And if you plot all these brains out with their measurements, they form this scaling factor, which is where you compare brain size, which is a certain scaling of the size, versus this tangential expansion, which is this basically an expansion of size. And so you see that this Gerification index increases with the expansion of the sheet and with brain size, and you get this characteristic regression function that's curvilinear. So you can actually characterize it in terms of a mathematical formula, and other brains should fit on that trajectory. So this is actually an interesting point, because this means that it's not necessarily a matter of this is a little bit different than the phylogenetic analysis, because it's basically a matter of the size of the brain, the expansion of that cortical sheet, and we see this increased Gerification. So it's a mixture of physical factors and genes that are controlling this process. And we can see that isn't necessarily, part of any one lineage. It's part of the mammalian sort of developmental milieu, if you want. So that's basically it. So we see in general that larger brains also have a larger GI value. We have this allometric scaling that we can derive in mammals, across mammals. And so that predicts growth of this sheet. It also predicts the conditions under which growth and Gerification occur. So we have this third study here where they actually took cortical sheets and they simulated them. They simulated their growth, and they simulated, more importantly, their folding. So you see that you have these layers here that form the neocortical sheet. You have these models of brains where you have a smooth brain, and then you start to have this growth of the neocortical area, and it starts to wrinkle. And this is these simulations are based on purely physical factors. So what they did was they took MRI data for brains from different stages of development and different sizes, and they made 3D printed models of these once they modeled them in the computer. And then they were able to model some of this folding, understand what the folding looks like in 3 dimensions, and show how, as the neocortex grows, this folding increases. In their simulations, they didn't account for any sort of genetic background. These are just images from MRI data sets. So it's just basically the anatomy and what's going on in the anatomy, and then modeling that with a computer and modeling that with a physics engine, to be more specific. So the physics engine is modeling this folding. And so this is a purely physical process. And so we find that, like, if you do this, you can actually replicate what the brain looks like during this process, what this neocortical sheet looks like. You get the same type of folds, you get the same type of behavior. And so this is maybe evidence for this physical hypothesis. But we do know that genes control this. We know that there's a whole family of MCPH genes or loci that lead to, if you have mutations in those genes, you have developmental mutations that affect the size of this cortical sheet and thus the folding. We know that mutations in this family of loci leads to microcephaly. I misspelled that there, where this mature brain remains small and lacks gerification. So if you look at like people with this developmental condition, they reach adult and they have very small brains, and those brains are smooth. They have some mental, they're mentally disabled as well. So that size of that sheet is, as it scales up, it has to scale up in size. If it doesn't scale up, someone's cognitive abilities are impaired. So this is some evidence that genes are controlling this process, and that it's not just a physical process. But you can simulate these with different physical tools. You can see that the fold wavelength is stable despite the volume. So you can change the volume, and these folds are stable. What I mean by fold wavelength is if you go from point to point on the surface, you get folds at a certain frequency. So you don't just get like folds at any frequency. There's a no matter what the size of your cortical sheet, those folds always come out at a consistent frequency, meaning perhaps that there are different places where there's a propensity for folding, or that the folds just happen because the sheet is of a certain, the material is of a certain type that it just folds in that way, or what. If we go back to our example of a flat piece of paper and we crumple it up, there's no evidence that crumpling is consistent over crumples. If we just take a bunch of sheets of paper and crumple them up, and we measure the folds and the fold wavelength, I don't know what the answer would be on that. I don't think anyone's ever done that. And that's kind of one of those Ig Nobel experiments that you could do, where you like just crumple a bunch of pieces of paper, spread them back out, and measure the fold frequency. But if you do that, I can't imagine that it would be systematic like this. So that might suggest that there's another role for genetics here. So this again shows that you have this mammalian ancestor, you have some transitional form, and then you have, in extant species, you have all this variation. And there are a lot of different things going on here. So it's, you have increases and decreases in brain size across the tree of mammals. You have increases and decreases in this Gerification index. And there are a number of factors that lead to that. There's one study that suggests that there's differences in cell proliferation that affect this. So, for example, when you have an increase in brain size and an increase in Gerification index from this mammalian ancestor, which is like what we see in humans, we have an increase in something called BRG and an increase in TAPs are present. So what are BRG and TAPs? So BRG is this basal radial ganglion precursor cell type. So these precursor cells are increased in their proliferation during this process. So from the mammalian ancestor, we have an increase in this certain type of precursor cell that proliferates in that cortical sheet. And that drives the size increase, and that also drives folding. But we also have these TAPs, and so TAPs are transit amplifying progenitor cells. And so these are another class of cell that is, when it's present, they're able to proliferate quickly and convert to different cell types. And so this might have something to do with larger brains being maybe smarter, or having enhanced cognitive capacity, as opposed to species of smaller brains like rabbits. And so you have different cell types, like there's very specialized cell types in the cortex aside from the typical cell types. And so some of those cells might actually play a role in other things that are going on in these bigger brains. So that's the kind of thing. We have just increases and decreases in those different factors. We also have to account for changes in cell cycle. So when cell cycle decreases in its frequency, you get smaller brains. When cell cycle increases in its frequency, you get larger brains. It's just how many cell cycles can you have during development. If you have fewer, you have fewer cells. If you have more, you have more cells, and that drives decreases and increases in this sheet, this cortical sheet. Similarly, you get an increase in brain size, a decrease in Gerification index in some cases, and that also leads to a different kind of brain. So this brain on the bottom is where you've increased the brain size but decreased the Gerification index. And that brain is going to look very different from a brain like a human brain with a lot of Gerification. So this is, I guess this is still an open question. I have some references here. If you're interested, you can explore this further. You can get into some of the details. It's a really fascinating area. It's a tough area to kind of understand, but once you get into it, I think you'll see that there are a couple of competing hypotheses here, a couple leading hypotheses, and maybe we can talk about this later on. It definitely has a lot of relevance to development and developmental differentiation that we talk about often in this group. So next thing I want to talk about is the GSOC for this year, and this is our one of two of our projects. So we have this graph neural networks project, and we also have the project on axolotl, building that axolotl atlas, which is called the Digital Microsphere. So it's like we have this specialized microscope that takes images from 9 different angles, and we're going to have data from these different angles, and they all take just different sides of an embryo. And so the idea would be to stitch those together into some sort of atlas or some sort of visualization. But this one is, I think, this is the one I've had inquiries about. This is the graph neural networks as developmental networks. So this maybe takes a little bit of explanation as to what we're looking for here. So here it goes. We're kind of played around with networks in this group. We've played around with neural connectomes. We've played around with gene regulatory networks, what we call interactome networks, and anatomical networks more broadly. So we've thought about all these processes going on in development, and where we're thinking about networks where a bunch of different components are connected together. And so these could be gene regulatory, these could be cells, these could be at the level of tissues. There are all sorts of ways you can look at these networks. We get the data for these networks from microscopy images. We get them from experiments, genomic experiments where people are looking at gene expression. We're looking at microscopy where people are looking at cells and tissues and their spatial organization. So we have all this data, and we have these network models. And so there's one other thing we do in this group, which is look at deep learning and machine learning. We also look at modeling, but that's not part of this. And the deep learning and machine learning work have been largely done in the service of extracting data from these data sources and sort of giving us a better sort of way to segment those data and to find salient features in those data. What we're doing this year is a little bit different. We're applying something called graph neural networks to this sort of analysis. And so this requires the person who's doing this project to create graph embeddings. These graph embeddings are, they should resemble, instead of resembling like a deep learning network where it's sort of this connectionist monster where you have like different layers that are fully connected and then you sort it out through weighting the different connections between the nodes. You're actually building these embeddings that maybe look more like one of these networks that we've been working on, these developmental networks. So they're not like typical, all connected networks. They have this structure. And so the structure is actually quite important. It tells us a lot of things about, centrality, about modularity, about things that, we talked about in different meetings and actually you care about when you want to understand development. So we're actually interested in building some of these graph embeddings. So the first part of the project will be defining graph embeddings, something that's computable, but also something that resembles development. So we want to use, biological rules, or we want to use ideas from like differentiation and differentiation trees, which we've talked about in the group, and other sources to generate different types of, say, like, things like movement output or developmental output. So, maybe even like a model of cell proliferation would be acceptable. They say movement output in this description, and it's a little vague. The goal, though, is to create a library of these graph neural network embeddings that we can use to simulate using a set of tools. So we can use PyTorch, we can use TensorFlow, and then I think next I'll talk about Towhee, and then you can see that you can also use something like that as well. So you can use a tool like that. You can create these embeddings. You can then simulate those graph neural networks using some algorithm, and then you end up with this really nice model. We work with simulation models in the group. We work with cellular automata and other types of simulation. So those are pure simulations. Sometimes they don't connect very well to the actual phenomena or the data. Like, for example, with a cellular automata, we can simulate pattern formation, but it may or may not resemble what's actually going on in the embryo. It just looks like it. And so we want to actually have something that is a little bit more connected to the data, that looks like it's something that's developmental. It could form patterns, it could move, it could proliferate, it could divide. The visualizations are created from the simulation. And those visualizations have different properties. They have to look sort of like they're developing. But that's not enough, because we want statistics that go along with it that show that they have these sort of properties of a underlying system that's doing these sorts of developmental things. So then, you'll join our group, you'll attend our weekly meetings, you'll give us reports on your progress and so forth. We have other, things that we've worked on in the past. So as the successful candidate for this position will contribute to the DevoWorm platform, which is this platform where we're doing a lot of pre-trained deep learning, DevoWorm AI, where we have other types of algorithms that one might use. They're not pre-trained models, but they're segmentation models and other things that might, we'll add to that library. You'll be a part of that library. But you can also, you can also make connections between these graph neural networks and some of those earlier models. And these are standard, some of these are standard, machine learning and deep learning models. Some are pre-trained models. The DevoWorm platform is a pre-trained model for C. elegans, embryogenesis. So there are a lot of opportunities here. You can also tie this into neural cellular automata, which is another thing that we've been working on in the group. So there are a lot of possibilities. The way I approach GSOC is to let people propose a solution, and then we evaluate the solutions and see which one is best, which one is most tractable. And then, that gives people a little bit of latitude to embrace what they're comfortable with, but also kind of get outside their comfort zone a little bit. But it allows them also to do something that they can actually accomplish. And so it's actually quite a good way to do this. We would like for you to have some basic knowledge of biology and complex networks theory. Now, knowing that not everyone has that, that's not a requirement. You can learn that in the group. But you definitely need to be able to program these kind of models and build them and have a familiarity with it. So that's the GSOC project. That's the Graph Neural Networks project. And we'll be talking about that more in coming meetings. So the next thing I'm going to talk about is this Towhee. And this is something that Krishna Katyal has been involved with. And he was as a contributor in the group. Last year he did a lot of stuff. He's been less active recently, but he's working now with Towhee. And this is a group that is trying to build machine learning pipelines for people who want to automate their machine learning process. And so they're looking at this X2VEC, which is an approach that originally comes out of text-to-speech or linguistic generation. So it's generating language. It's a language model type thing. But there's this idea of anything to VEC. So Towhee allows you to automate this process of taking anything and turning it into a vectorization which then allows you to build a machine learning model from it. So they're doing this, they're helping you encode unstructured data into embeddings. And so this would actually be quite useful for the Graph Neural Networks project, if one decides to do that. There's documentation here. There's a list of pipelines. So they're on GitHub. You can start their repository here. We have out-of-the-box pipelines. So there are a number of out-of-the-box pipelines one can use for different things. They're not all, one area. They're not all like NLP, or they're not all like image processing. They have different things going on here. So yeah, there are a lot of embeddings or pipelines to choose from. There's documentation that gives you sort of this idea. It basically aims to democratize anything to VEC, allowing everyone from beginning developers to larger organizations to train and deploy complex machine learning pipelines with just a few lines of code. So this is something that you can build atop PyTorch, scikit-learn, and OpenCV libraries. So this works with other libraries that you might want to use. And so yeah, this is great. So this is something that they're applying for GSOC students as well through our organization. So our organization is the Orthogonal Lab, and so if you're familiar with that, we have a list of projects through there, and it includes, this year we're applying to be an organization, including projects from Orthogonal Lab, which is an AI ethics project. We're applying through DevoWorm, which has the 2 DevoWorm projects. We're applying through Rockwire Community, which is another group that does a lot of smart communities and mobile app development. There are 2 projects there, and then there's this Towhee project. So definitely check that out if you're interested. The difference between the Towhee project and the GNNs project is that the Towhee project is more focused on these pipelines, building pipelines and quality control and things like that, whereas the GNNs project is DevoWorm oriented. You're going to be working with biological data, but you might use things like Towhee or some other tool set or some other libraries to accomplish that. So that's what we have there. I wanted to talk about this blog post because it links into what they're doing at Towhee. And so this is transform anything into a vector. So I talked about anything to VEC. What does that mean? There's this approach called entity to VEC or anything to VEC. This is using cooperative learning approaches to generate entity vectors. And so this is by this author here, works at IBM Watson. So businesses commonly need to understand, organize, and make predictions about their users and partners. So they're maybe doing things like churn prediction, which is predicting users and their turnover, or different types of advertising partners. They use an example of clustering. The challenge comes from trying to represent these entities in a meaningful and compact way to feed them into a machine learning classifier. So that's what they're doing when they create these embeddings. They're creating basically data structures that allow you to represent different entities. So in developmental biology, we have a lot of entities at the cellular level, at the molecular level. We're probably interested more on the cellular level in these GNNs, in these embeddings that we're asking for in the GSOC project, but also combining the 2 scales, genomics and cell biology, is also it's just a matter of finding the right data that matches up, and aligning data is very challenging. So maybe we don't go there first. Maybe that we save that for later. But so this goes back to natural language processing. There was a team of researchers at Google that created something called Word2Vec. And Word2Vec, and this is the preprint here, which is a technique that represents words as continuous vectors called embeddings. So what they did in Word2Vec is they created words that were continuous vectors, these embeddings, and then they would embed them in this dimensional space, and they'd find the distance between these words. So for example, you have a man to a woman, a king to a queen, walking to walk, and swimming to swim. So you have these distinctions between male and female, you have these distinctions between verb tense, and then you have this distance basically in this 3-dimensional space of you can't do that. You can do it, I guess, by looking at its Levenshtein distance, which is the distance between the characters in the word. So walking and walked, you have a certain number of letters that are different in those two. And so that gives you a distance. But there are other ways to calculate distance based on context, based on other things. And so I think they use a Levenshtein distance in the word DeVec, which is just like a simple, it's not a Hamming distance, it's like a Hamming distance. It's a specific type of sort of Hamming or diff type distance. And then you also have correspondences. So you correspond a country to its capital, and you can draw a line between them. So these are how embeddings work in the linguistic example. You have all these things that are going on. You figure out which terms or entities are closer to other entities, and you build a network from that. Or they just show the linear relationship, but you can actually build networks from this. So it's kind of an interesting way to approach it. So word DeVec has been a staple of NLP, or natural language processing. But now we want to take that outside of the linguistic context and make it available to other types of data. So they talk about business in this post. I thought it was nice because they kind of talk about it very simply, but we're kind of interested in biology, and biology is quite a bit different because you don't really have, you don't really have words, you need words, but you want your objects of analysis are maybe more complex than words, or like customer databases. They're often like cells or identities or gene sequences, but they don't exist in isolation. You have to express them. So there are all sorts of like different entities and associations we can make. And the key is to get in. There's a great complexity there, like there is in language. The key is to find a subset of those and to build these representations. So here's an embedding for words. You have source text, you have training samples, you have, they kind of go through this where they take each word in this sentence and they train, an association of Brown with different words. So, what do you associate Brown with in this sentence? You associate with all the other words in the sentence. And so you train the model to associate, say, one word in the sentence with every other word in that sentence. And that'll allow you to build a model that suggests that sentence is likely. So this is the way you build these types of things. You build it from a corpus of data. So in this case, they had 100 billion words from Google News and building word DeVec. In the case of our data sets are maybe smaller than that, the ones at least we're going to work with in the project, but there's a lot of biological data and potentially even more than this, depending on how you define the entities and how much data you can actually transform into something that you can get associations from. So that's why I'm connecting it with all this other work we've been doing on extracting data sets from microscopy data. There are possible genomic information we can use. So there's a lot of potential, and there seems to be a lot of, these seem to be pretty large models, these embeddings, but, our embeddings don't have to be that big. They can be rather small. And, we don't know what the performance is going to look like, so I don't want to say anything about that. But, this is a lot about word embeddings. There are different strategies of this. And so I use this as an analogy to what we're doing in developmental biology to say that this is what's possible, but I don't know how it would actually work. So this talks more about word vectors and how you make these associations. You have an architecture of these embeddings. You have a person embedding, a single dense layer, and an output. So you have this person embedding, which is like you give it a person, you have a database of people, and you have it try to understand, so this is a picture of Barack Obama. Which embedding fits Barack Obama? And you have the labels here, but the machine doesn't know what these labels are. It just knows that they're different embeddings. And so it just tries to match up this image with the embedding, or like the residual network that comes from training it on images of Barack Obama or Hillary Clinton or Bill Gates or whatever. And so you'd be able to differentiate between those images by its statistical properties. And so that's what you're trying to do. You're trying to take any one sample, fit it into an embedding, and then see which embedding matches it most closely, and then that's the embedding you would use. That's how it recognizes these things. So that's the dense layer and then the output. So there are different methods in linguistics. They use entity DevEc, they use pre-trained word DevEc, bag of words. That's most frequent. These last two are older techniques and they're not as good. These are the newer techniques and they're much better. But, these are, of course, reliant on a lot of data. So this is something to keep in mind. So you can do this with the Yelp data set as well, where you look at different recommendations and you try to fit it to you look at different retail establishments and then you fit them to this output. So yeah, you have to, it's another classification task. So then, this is basically anything to vec just simply means taking anything and turning it into a vector that you can use in machine learning and then using a pipeline to make it into, turn it into an embedding and then use a pipeline to turn it into some data that we can use or some set of classifications that we can use. Either way, it works out. So next thing I want to talk about is the special issue that we're working on. So this is between myself, Richard Gordon, and Susan, I think. Yeah, she hasn't committed to it yet. But this is a special issue of the journal Cells. This is something current research in developmental biology. And the thing we're trying to do here is we want to create an issue on biological networks, but very broadly conceived. So we're interested in biological networks that encompass a number of different areas, including soft materials and what's going on in the physics of embryos. So this would include things like tensegrity, which we've talked about previously, or other types of physical networks where you're looking at physical forces. They could also be other networks that are, like networks where you have divergent function, things like that. So there are a lot of different, I have to think, I haven't written up the description of what this is going to look like, but this is what I'm thinking. And so I think this will be a nice special issue. Hopefully people want to help with this. You're definitely welcome to join in. Let me know. And this is something longer term. I don't know. There's a call for a special issue from this journal, but we might not go with this journal. We might go somewhere else with this. But this is definitely a nice topic, and we'll have to work on that. So last point I want to make today is I want to talk about readings for today. And we have a lot of things in our queue here. So why don't I talk about gene regulatory networks in different ways? And so what I mean by this is that there are different ways to represent a gene regulatory network. Like we talked about with the embeddings, you have these images and you're taking like the or words and you're taking the features or things you think are important and you're putting it into this model. And then you're using that as a way to kind of analyze different images. In gene regulatory networks, people are modeling these things in terms of what they're observing from the natural system. So they look at things that they think are important. Then they generate a model. It could be a computational model or a phenomenological model where they look at just what's going on. And then they see how it predicts the data. So I have these two sections from the Eric Davidson book. So this is the Eric Davidson book. There's chapter 1, which is regulatory hardwiring. And Eric Davidson developed a lot of nice models of gene regulatory networks. Not a lot of those are actually computable for a number of reasons, but he did a lot of the early work on this. So this is chapter 1, regulatory hardwiring, a brief overview of the genomic control apparatus and its causal role in development and evolution. This just kind of goes over like gene regulatory networks and how they've sort of evolved in the tree of life, but also how it play a role in development. And so there are a lot of things we consider about like genome size and different modular structures for these networks. So in this case, we have a network where you have two different modules that are being turned on and off. You have different features. So these modules consist of spatial repressors, meaning like where in space some gene is going to be regulated, upregulated or downregulated, so where it's going to be turned up or down. They're different signals from adjacent cells. So if you have another, this is in a single cell. If you have another cell as your neighbor, it's going to send a signal to tell you to turn down your gene expression or turn up your gene expression. Cell cycle control. So like as I said earlier, cell cycle has a certain frequency, and if it's higher, there are more cells produced in terms of proliferation. If it's lower, fewer cells produced. And then different lineages. So the cell might change its lineage. It might branch off onto a new lineage or be a lineage identifier. So all these things are important for, and as we know from even the C. elegans example, a lot of these are important in the developmental sort of structure of how the length of development, how things unfold, and so forth. You have two modules in this example. And these two modules actually can be controlled by an OR gate. So you can have two modules that have settings. They might be, deterministic settings. They might fluctuate. You can then have this output here where there's an OR switch. So you either use module 1 or you use module 2. You can also have an AND gate or an exclusive OR. So you can have these logic gates here where it turns module 1 and module 2 on and off, or combines them, or something like that. And then this results in an output, which is RNA. So your DNA is transcribed into RNA. These regulators move across the DNA, the putative DNA. There's no DNA in this model, but this is a cis-regulatory element which sits right directly upstream of the gene or the coding region, and then it basically decodes that DNA into RNA using these different factors. So it might produce a lot of RNA, it might produce a little bit of RNA, it might produce, modified RNA depending on what the regulatory machinery is doing. So that's one way to look at it. There's this other model where you have modeling gene regulatory networks using neural network architectures. So in this paper, they talk about gene regulatory networks encode the complex molecular interactions that govern cell identity. They propose in this paper a method called deep SEM, which is a deep generative model that can jointly infer GRNs, or gene regulatory networks, and biological meaningful representation of single-cell RNA sequencing. So this data is scRNA-seq. This is a next-gen sequencing technique where they're trying to represent these type of data. I guess they're trying to replicate this type of data. So this is like where you have not only the sequence of a transcript, an RNA transcript, but you also have an amount that's generated. So you have a certain amount of this, number, a copy number. So again, if it's turned up, you're going to have a larger number of copies. If it's turned down, you're in a smaller number of copies. They're all going to be more or less the same sequence. The thing about transcription is it produces transcripts of different length and different sequence because it's taking, different parts of that coding region and transcribing it into RNA. So it's going to look at, they try to align these sequences up and find like the sequences in common and then say that that copy number is high or low of that alignment. That's basically how these next-gen sequencing technologies work. In particular, we developed a neural network version of the structural equation model to explicitly model the regulatory relationships among genes. So a structural equation model is a statistical model where they take a different number of different variables and they look at their relationship. They use some statistical tests to assess. So it's like almost like a network of different variables and how they're all related to one another. This is often something they use in multivariate, a case where you have a multivariate data set where you have a lot of potential causal factors. You don't know which one, what causes what, so you try to sort it out using this type of model. So this helps us with regulatory relationships because we want to know what came first and what came second. In terms of genes, just looking at genes, we don't know which genes activate other genes. So a lot of these gene regulatory networks are like one gene that is transcribed, and then that RNA transcribes another gene by activating its regulatory machinery. And then that in turn activates some other genes. So you actually have these hierarchies in some cells where you have some function like muscle. If you want to go from like one type of cell to another, I'm going to go to my board here. You might have a network of genes, so you have like say a single gene here that's an activator, and it might activate a bunch of genes down here. So like if you activate this cell here, or this gene, it activates these, it turns on these other genes by virtue of its RNA production, or its mRNA production, I should say. It's messenger RNA that activates the transcriptional machinery here, the regulatory machinery, turns these genes on, and then when they're turned on, their mRNAs in the cytoplasm, it's signaling, but it's also turning on these other genes down here. So you can see that, like, if you want to create a muscle cell from a stem cell, you can use a factor called MyoD. You can introduce it into the cell, and it will turn on this MyoD gene. It'll activate it, activate its regulatory machinery. It produces a lot of mRNA. And this mRNA then will activate these genes down here. It'll activate the regulatory machinery and then the gene. It'll produce a lot of mRNA copies here, and it'll go on and activate more genes. And they call this a gene activation cascade. So this is a nice example that's not too complicated. But suffice it to say, it isn't that simple. There's a lot of cases where it's not this simple. It's not hierarchical, for example. It can be very, the causality can be very hard to follow. And, this is something we've talked about in this group and in my other group, where we have, where we have these reciprocal causality situations and all these other things. So don't think that it's always hierarchical like this. In this case it is, but in other cases it's not. So they have to use these different models, structural equation modeling and deep learning, to kind of get at some of this. So they use the deep generative model to generate these conditions, these GRNs and some of their products. And then they try to use the SEM to figure out which genes are causing what. So benchmark results show that deep SEM achieves a comparable or better performance on a variety of single-cell computational tasks, such as GRN inference, scRNA-seq data visualization, clustering, and simulation, compared with the state-of-the-art methods. In addition, the gene regulations predicted by deep SEM on cell-type marker genes in the mouse cortex can be validated by epigenetic data, which further demonstrates the accuracy and efficiency of our method. Deep SEM can provide a useful and powerful tool to analyze scRNA-seq data and infer a GRN. So again, you're using this deep learning component to generate these models of GRNs, and then you're using the SEM part to analyze them and figure out how they're structured. So it's a nice paper. It's very, it's, again, like, this is very hard to understand because they're using, not only are they using a lot of the genomic terminology, they're also using deep learning and statistical terminology. So you can see that, you have these transcription factors here that serve as hubs. They target different genes and their activation. And this is basically how a gene network is structured. There is typically this sort of semi-hierarchical, at least a semi-hierarchical nature to it. You have these hubs that activate a bunch of downstream genes. They can be strictly hierarchical, like in the MyoD example, or they can be like this. And this is more kind of like a scale-free network with hubs. So this makes it look, a little bit more like, it's a little bit more decentralized, but then again, it's also, there's a lot of, direct control here. So another example I'm going to use here, or draw from, is this paper that came out, I think recently, very recently. It's a gene regulatory network balances neural and mesoderm specification during vertebrate trunk development. So this is a nice, very long paper. It's nice, but I want to go through some of the highlights. So again, they use a single-cell RNA-seq technology. It reveals a signature of neuromesodermal progenitors. So these are types of cells that are sort of in that mesodermal layer the embryo, but they're sort of have this neural precursor property. These are progenitor cells. They're able to find this with the single cell RNA-seq. They look at these in vitro NMPs resemble and differentiate. These are the neuromesendermal progenitors. Resemble and differentiate similarly to their in vivo counterparts. So they're doing this in I guess it's an in vitro model. So neuromesendermal progenitors generate cells of the spinal cord and somites. So they're in the embryo. They're not quite forming any tissue yet, but they're going to go on to form cells in these areas. And they're doing this in vitro, so they're trying to match the in vivo results. And so this is, and so they want to investigate how the generation of these precursor things like neural and mesendermal cells, they generate these tissues, and then they generate the orderly extension of the embryonic axis, which is this anterior posterior axis where you have the spinal cord and you have the axis of the body from head to tail. So there's also a dual role for retinoic acid signaling in NMP induction and neural differentiation, and then transcriptional network regulates neural versus mesendermal allocation. So this is in a biological system. This is where they look at the network and they see what it's doing during this process of differentiation. These cells, as precursor cells, they self-renew, and when they differentiate, that self-renewal is gone, meaning that they can live forever in that state until they differentiate. So they can keep dividing and not, experience any degradation, whereas in cells of a specific fate, they typically live a certain number of divisions and then they die. They apoptose. And so this is an important switch that gets turned on and off in these cells. And so they kind of talk about the cell type, and then they try to discover the gene regulatory network that's involved in this. I'm trying to see if they have a figure where they show this network. They show a lot of things about the different, they show the molecular signatures. And they actually show a little bit of the signaling cascade and maybe some of this network. So this is a route from this precursor cell lineage to these different other types of cells, as neural lineage cells. So the PSM is, I don't know what that is. NPC is the neural precursor cell. So this is a neural precursor. It's outside of this germ layer, and it's now on its way to becoming some sort of neural cell. So and I don't think I'm going to go in and try to find a figure of this. Just suffice it to say that this is another method of looking at gene regulatory networks. You can actually look at the concentration of the products during different periods of development. So that's a nice way to look at the activation of these networks over time. The final paper I'm going to talk about is this evolving differentiable gene regulatory networks. And this is Doug Wilson and Sylvain Coussat-Blanc and Kyle Harrington. So these people have been working in the artificial life community and on gene regulatory networks in development. So over the past 20 years, artificial gene regulatory networks, so these are artificial computational models of these type of networks, have shown their capacity to solve real-world problems in various domains, such as agent control, signal processing, and artificial life experiments. They have also benefited from new evolutionary approaches and improvements to dynamic, which have increased their optimization efficiency. So in this paper, they present an additional step towards their usability in machine learning. So they've actually talked about these type of networks in previous papers. This is a newer paper where they talk about differentiable GRNs. So they're implementing these GRNs that differentiate. And so they actually can be optimized using stochastic gradient descent. So they use a standard machine learning benchmark data set. They evaluate these networks and how they can be optimized. So in biology, they're not always optimized. And the difference here is that you can create these networks that are inspired by gene regulatory networks. And I think in the last paper you saw how messy that can be in a computational model that can be optimized in terms of gradient descent and that sort of thing. So they're not using any sort of genetic algorithm here. They're using gradient descent. And so artificial GRNs were first proposed using a binary encoding of proteins with a specific start and stop codon, similar to biological genetic encoding, which was like about 20 years ago. GRNs have since been used in a number of domains, including robot control, signal processing, and wind farm design, and reinforcement learning. So this is a biologically inspired model that's been used for a lot of other applications. And they've been used to investigate a number of questions in the context of evolution. So one such question is the relationship between their robustness against noise and against genetic material deletions. Redundancy of genetic material was proven to enhance evolvability up to a point, and it was shown that modular genotypes can emerge when GRNs are subject to dynamic fitness landscapes. So these networks that we saw in the first example in the Davidson book, they've sort of formalized these a little bit, and then they're using them to simulate these types of things. And they can generate these types of behaviors that look biological, but they're actually not, they're not biological in the organic sense. They look biological, their outputs do. So in this paper, we present a model that takes the process of optimizing GRNs one step further by introducing the differentiable gene regulatory network, or one that's capable of learning. They show that differentiable GRNs benefit from a combination of evolution and learning. You can use modern platforms like TensorFlow and GPUs, and that should be able to help you make these networks all the more powerful. And so that's what they're trying to do here. And so the first set of GRN models weren't necessarily very good, but these are better. That's what their point is. They're able to actually show some really interesting effects, like the Baldwin effect, which is where learned and adaptive behaviors have a fitness advantage that can facilitate the genetic assimilation of equivalent behaviors by subsequent generations. They also can model plasticity as an adaptive response. So yeah, they just show this differentiable GRN model and they walk through it. And I don't know if they have any nice figures here. This is a nice figure. A GRN layer model is a composition of other layers. So here you have inputs, you have outputs, and you have regulatory units. And then you use the softmax function. This output mask gets fully connected. And that's how they do this. So they're able to get like these, they're roughly like you have these, what you might consider to be genes. You have an input, you have a recurrent state, which is a feedback. You evaluate it using a softmax criterion, and then you have this output, which is the output mask, and then that gives you your answer. And so you can use this on a number of, non-biological problems. So this is, again, another example. A GRN layer model is a composition of other layers without protein concentration normalization. And so the protein concentration is, where we have the switch that goes on and off. And, if you use a softmax, I guess you get like this concentration gradient that it has to be normalized or not. And so you end up, that's how you do that. And so, they have some nice graphs of before and after training, so they clearly learn and they clearly minimize their error. And so that's that. So that's our, I think that's all I'm going to go over with you for this week. I just wanted to show you the different versions of gene regulatory network that you can have. I know that we've been talking about this in past meetings, so I think that was a nice overview of that. So next up is Karan and some of his materials. And after he's done, then see you next week.
Yep. Is it visible?
Yeah, I can see your screen.
Yeah, I was going through 2-3 papers. They were kind of based on, gene expression and, how they were using pictures to sequences, and they were using it to, predict like abnormalities or things like that in the pictures, and trying to find a connection between the two. Like if you have a gene sequence and you have a picture associated with that, so they were trying to find, like the neural networks being trained on both of those things so that it can, find out similarities and things like that. So there like 2, 3 pre-trained networks that are already existing. So I don't know which paper exactly dealt with the accuracy and all those things. So transformers were again, a big thing that we used, I think.
Yeah.
Uh, this one. This is this not that's maybe the other one. This maybe blog has something similar.
So this is from DeepMind.
Yeah. This one's from DeepMind.
All right.
They come up with a lot of good stuff. So yeah,
Yeah.
it's a good source. Like their blog and their research papers that they publish. Uh, uh, I don't think this is the exact one, but it was something similar. So they were, talking about the accuracy models that are there, and how usually for mammals, like mostly mice are the most studied uh, thing. So they had a lot of mice-related data that was there, I think. So they trained it on that, and I think it was kind of uh, like it could be used for a lot of things. That's why I was, trying to find it.
Right.
Yes, pressures. They're not keep it this or English is pretty
So it
good.
looks like they're trying to do something called site prediction, if I'm not mistaken, where
Okay.
they're looking on the gene and they're trying to find different
Okay.
parts of the gene. Yeah.
Okay. Uh, is it element? Maybe somewhere here, I think, the people. Um, yeah. So, also regarding like gene sequences, the epigenome is like it contains the actual transcript of how it doesn't work that way yet. The epigenome thing, like if you have a gene sequence that is there, the epigenome will tell you how it's going to encode proteins using that gene sequence. Uh, like
Yeah. Yeah. I guess
I
it's,
think
yeah. So it's, you have your gene, which is your DNA sequence. And
okay,
then there's a method of transcription. So there's a transcriptional machinery that
okay,
hooks onto the sequence and transcribes it to RNA. Okay, so it's going down the gene, and there are a lot of different
okay,
marks on top of that, like in the gene, in the promoter regions, what they call it's just before the main gene, that tells it what to do. So you have, different promoters and enhancers, and they kind of, tell the transcriptional machinery where to go and what to
okay,
take transcripts of. And then you also have these histone markers, and these are sites where they either enhance the activity of a certain downstream component, which is down from that histone mark, or they turn it off. So there are ways that there are a lot of different ways that the gene gets expressed through a lot of the epigenomic
okay,
stuff
okay,
and transcription. So you have a gene sequence, but you end up with something quite different as the RNA, and that's what ends up
okay,
being made into the protein.
okay, okay, okay. Uh, okay. I think. Hmm, okay. I think using it to transform it makes sense. Maybe it was this one. Full text. Yeah. Hmm. Okay. Mm-hmm. Genomic track. They've compared like three of them. So I was kind of thinking, doing maybe a demonstration of something similar, but for a different species, maybe if I had the data set or something like that.
Yeah, I think
Uh,
that. Yeah.
because there are a lot of like base transformers, that you can tweak it with to get different things done with them. Uh,
So
you know,
in this paper,
yeah,
they're using
yeah.
something called an N-former. So what is the difference
Okay.
between that and a transformer?
End former, I think it depends on the where you're implementing the transformer mechanisms, for something that is very custom made like this. Like gene expression is more of a, I think there are a lot of probabilistic mechanisms involved, I think, in this particular case. So I think you have to incorporate like different kind of, methods to make the algorithm work properly. I think it's that. There can be other reasons. Otherwise, end formers, it's not like, there's something special or they're vastly different or something like that. And formers, I think it's a pre-processing transform or something like that, I think.
Okay.
Yeah. Otherwise, DSS. Genomics. Yeah. Okay. Data set in this. Let to find where I found the data set actually, because that had more information. This is the transformer paper as mentioned.
Yeah.
That's it, data set. Actually, nothing. Nope. This is regulatory rates. Okay. Okay, sequence from one analysis only. Chromosome architecture. So the powerful assays. Body weights. Package from that. Okay. Sequences. They're using DG package. Okay, maybe it was this one. Genomic pages. We need to reference manual. Description. Okay. Okay. Straight. Expand area. Okay.
ENCODE.
Got it. Yep. Variant was this one. Data. Info project.
Yeah.
Okay, nature. We'll have to work that thing. I'll have to check maybe that it's free available. Is this it's available maybe?
Yeah, the ENCODE project. Yeah.
Yeah. Yeah, I think maybe it was this one.
Okay, yeah, that's
This was Genomics. Core. Yeah. Something like this, yeah. Okay. This must be low. See, Elegance is also there.
yeah. They even have organoids in there, I see.
Yeah. So this. Okay. Van moet ik zeggen, DNA sequence met chips zijn. Body plus RNA sequence. Maybe something related to develop the motor research.
Yeah.
I think there was some option of coordinating and comparing data sets.
Right. That's
So this. Maybe. Okay, organs are there. Analysis, genomes, and the Phenotypes. This overrest. Yeah. IDF. Two. Target. So decent amplification. Okay. Gene expression. Sure. Okay. Is this like, could we add some sort of a feature maybe with the axolotl data set? No, wait. I don't think that will be viable with this.
You mean a genomic component?
Yeah, like maybe because from the pictures itself, no, that would be.
Yeah. Is this axolotl or is this another organism? This is
This is Homo sapiens. Yeah, like I don't think they have
you
something
know,
for
yeah, I
axolotl.
didn't see anything there. I mean, we
Yeah.
could, and I mean, it would be kind of hard to, it's always kind of hard to integrate the two kinds of data, but it would be nice to have
Yeah.
something. I actually think there are like a lot of spatial expression data sets. I know that they have them for like C. elegans and maybe for zebrafish. I don't know. I don't know about axolotl. I can look into that and see if they exist.
Okay.
Because I know it's a common model organism.
Okay.
Yeah.
Axolotl.
Yeah, there we go.
No,
Doesn't look
I don't
like
think
it.
so.
Yeah.
Mighty.
I don't think that's not it.
Okay.
But yeah, they have like Drosophila and yeah, and C. elegans, the mouse, human.
Okay. Yeah. Maybe I'll have to just do a wild reference search, maybe with any kind of axolotl data. That's
Yeah,
there.
yeah.
And then try to do that. Yeah.
Okay, yeah.
Yeah.
But yeah, this is great. This is the they have that
Yeah.
combination of like deep learning and genomics, and that can break a lot of people. Like this is just like both fields are very intensely like technical, and they have their own terms. So
Yeah, very, very vastly different. But I've seen transformers. They're mostly used in applications where they're using both. Like they either using one and generating a prediction, and they're using the picture maybe to, verify or really confirm that that prediction is, giving us this. And they kind of going hand in hand, with that
yeah,
aspect. So that was there. Another topic that I was going to was, I think, again, hydrophobicity problem. Molecular, it has to be deeply rooted in molecular physics, though, because you have like physics model, that's checking out whether how those hydrogen bonds are actually working with the LND transformation.
yeah.
And kind of okay. So yeah, that was that. Just start working. Okay. This. So it's kind of like, it's trying to differentiate between the objects that are there. So I had to go through a neural network. It's kind of giving some weird results here. This was there. But for the 4D structure, it won't matter that much, all these things. It's just for an additional thing that you want. It'll matter. This was, I think, this is trying to differentiate how many, this is maybe a stage 4 or 5 axon embryo. So it's kind of trying to find out how to map that thing, and find out what all things are there.
Yeah. Yeah, that looks good. So you're segmenting the images and you're at different stages and you're
Yeah.
putting in. Okay.
So I have like pictures from all sorts of areas. It's just I had to find a way to show that in different stages that is there. So if I could have like a real count of the amount of times that thing is divided, and along with that, the axolotl stage that is going on currently. So I could put it on a timeline, like and what we can expect it to go like. Maybe if it's increasing logarithmically or linearly, I think it's going to be logarithmically only, obviously, but
Right.
yeah, something like that. But wait, the thing is, it still hasn't got to the stage where it can clearly properly differentiate between different uh, uh, sections in that embryo. So let's see. At least a general thing is more viable right now. I'll probably work on that now.
Yeah.
So yeah.
Well, that looks good. Yeah. Yeah.
Yeah. Okay. I think it's saved on my other drive, the actual pictures that are there. Susan had given a lot of pictures, I think, they're till stage 8, if I'm not wrong. I think find those interesting, and it saved them in my Ubuntu actually.
Right.
So, let's see. Okay. Yeah. Okay. I think we'll have to log into a different account for that. Yeah, I think this is it for now.
Okay. Yeah, that's good.
So,
Yeah. Well, that's good. Thanks for the update. And
yeah.
yeah, if you find anything interesting, you can put it in the Slack channel, or if you don't do it, you can come to the meeting, of course. But like, I know we're going to have GSOC students applying this year, or so.
Yeah.
That's
Okay. Yeah, I'll post any data set or something interesting, that I find.
okay.
Just like I'll do things with the encode thing at least. Yeah.
Yeah, I'd be interested to see what you can do with the encode, because that's always been like, I remember when they first came out, and it was like just so overwhelming with data that there's got to be something
Okay.
in there that people haven't really
Yeah.
thought about before. So
Overlook this.
yeah.
Yeah. Okay. Yeah. And okay. There are a lot of again. Yeah, DKs and these sources of this. Okay. Yeah. Let's see. Data sequences. Okay. Drive. Embryo. Where is it? Yeah, I found it. Copy one. Discord. Okay. Paste this here. This is audio. No. GR. Copy link. Yeah, this should be it.
Okay.
Yeah,
Okay.
this is the one.
Yeah, there it is.
So I kind of take it like stages, all stages, and try to feed them. So like this was maybe probably stage 6,
Okay.
which okay, 6 DSC 1007. 07. Okay, yeah. So it's kind of trying to find these lines here,
Yeah.
the amount of times it is divided on, so the thing that could work is, at least for an estimation purpose, would be if I could contrast them in a very, on different levels and, have it align all those layers and then tell, if you can navigate all any kind of lines that are there, here, or here, here.
Yeah.
Because it's just a big drastic change in the pixel uh, number that is there, right? If it's going like this, traversing like this, so the pixel levels will be, it's like kind of similar, the numbers that's going through, then there'll be a change here, something like that, maybe contrast different layers, something like that.
Yeah.
To at least find out how many times it's divided, and what one can we expect, like from a stage 6 as model. So it'll maybe give a prediction also, like it should have these many divisions by this time. It's kind of has, let's see, stage 7. I had some good pictures that I had chosen for this particular part only, but still was kind of giving stage 506. Okay. Stage 506. Okay. It's plus 006. Yeah, like this better than maybe. Okay, yeah. Yeah, no, it's okay. Maybe map it with 2, 3. Yeah, so H4. All these stages are pretty easy because I think they're not more than 2 or 3 divisions.
Right.
So, this was stage 7, so okay.
Oh.
Yeah, stage 7.
Okay.
Yeah, this is different. This is
Yeah. Oh, there it
pretty
is.
sure.
Yeah.
Yeah. Like finding anything, like at least an estimate would be okay. If you're giving it to a neural net, it'll definitely
Yeah.
get. I wouldn't expect it to work out of the box like that.
Right. As long as they're all the cells are consistent, like as long as you don't have gaps within the. Yeah.
So that is there. Guys, let's see. Stage 8. this might need some new samples, I think. Yeah. Okay. Yeah. Just 1 minute. Okay. Yeah, I think it's dead the other day. Then Amplifying. I think this is it. Kind of kept another folder, I cherry picked, some pictures that I knew they could give good results, but
Okay.
I think it's there somewhere else, maybe somewhere offline.
Yeah. Yeah.
Because this has a lot of pictures, like I've chosen like 3-4 per stage.
Okay, like that.
Okay. Let's see. I think we'll have to find it some other time.
Okay.
Because
Yeah,
it's
that's
stored
fine.
online.
Yeah.
Yeah.
Well, yeah, it's nice to see that, and I think it's great that kind of getting it together and trying different techniques.
Yeah.
Yeah.
Oh. Okay. Yeah. Yeah.
Yeah.
So yeah, let's see. And look into this gene transcription thing as well. Like, if that can be any sort of thing that anybody's done so far, anything similar that we added with the model itself. So let's see. Yeah.
All right. Sounds good. Okay.
Yeah.
Well, is that it for today?
I think yeah.
Okay. All right.
Things used to relate to that anything, but not playing. I couldn't find any clear answer to that. So that is still ongoing.
Okay. Yeah, that's fine. Yeah. All right. Thanks for the update. And yeah, next week I don't know what we'll do, but if you have another update next week, I'll try to look into some of the spatial transcription stuff. Maybe I'll present some stuff on that just to see what's out there.
Okay. Spatial transcripts. Okay.
Yeah.
And try to go through mine also, at least. Trying to really.
Okay.
So.
Yeah. All right. All right. Talk to you later.
Yeah. Okay. Yeah.
All right. Take care.
Okay. Bye.